Okay, so I've put off recording this because I kept thinking we'll be in person soon and I can teach this to you in person. But as the weather has continued to prove, my plans are not quite working out the way that I had hoped. So, with that being said, we're going to cover meiosis today using Ed Puzzle, and we'll review it the next time we're together in person, but I can only delay so much. So, we're going to talk about meiosis today. And the main idea is that meiosis produces haploid gametes. Now, there is going to be some terminology here that you are not super familiar with just because we've not really covered it yet. I've kind of gone out of order as far as the way that the book goes. Um, but there's been certain things that I think that you need to understand um, that the book doesn't quite put in the same order. So, with that being said, I don't know how many of y'all have heard of mitosis before, but mitosis is a similar process to meiosis, and mitosis is what goes into further detail. So we'll go into more detail as the year progresses, but just to kind of introduce and to cover the basics, we're going to talk about meiosis. So, before we get into that, there are a few things you need to understand with chromosomes and chromosome number. So, a gene is a segment of DNA that controls production of proteins. So, we talked a lot about DNA and proteins and all of that last chapter when we talked about transcription and translation. Human body cells have 46 chromosomes. They have 23 pairs, one from each parent. So, you'll hear people talk about 23 pairs of chromosomes, and that comes from the fact that we have 46, and you inherit one from your dad and one from your mom. So there's something called homologous chromosomes, and these are chromosomes that make up a pair, one from each parent. They have the same length and centromere position, and they carry genes that control the same inherited trait. So you'll hear um, hetero and you'll hear homo. So homologous chromosomes, homo means same. So homologous chromosomes are the same size and they have the same centromere position. So you may be like, okay, Ms. Brammer, what exactly does that mean? Well, here we have our 23 pairs of chromosomes. Now, if you see here, there's two chromosomes, so there's a total of 46, but there are 23 pair. Now, notice they are all numbered except for this last pair. The 23rd pair is known as your sex chromosome. And basically, what that means is that's what determines the sex of the human. But we know that these are homologous chromosomes, all except for the 23rd pair, because notice they're all the same length. Now, 2 is longer than 3, but both chromosomes found in the second pair are the same length. So these two are homologous chromosomes. So this is an example. Looking at this, you see these are a pair of homologous chromosomes, and essentially what they do is they determine the type of earlobe that you have. So notice this is an attached earlobe and this is an unattached earlobe. So if you pay attention to your earlobes, take a look at them and tell me, do you have an attached or do you have an unattached earlobe? And that is determined based off a pair of homologous chromosomes found within your 23 pairs. So the two terms that we have here are haploid and diploid. So haploid is having only one set of chromosomes, and gametes are haploid. So a gamete specifically is a sex cell that has half the number of chromosomes. A diploid cell has two sets of chromosomes. A diploid means having two sets of chromosomes, and body somatic cells are diploid. So, what happens here is fertilization occurs, and fertilization is the process by which one haploid gamete combines with another haploid gamete, one from the male and one from the female. Now, if you notice how you can remember something is haploid or diploid just by the name, or what that means is di means two. So in diploid, that literally means you have two sets of chromosomes. If haploid is N, and kind of incorporating math a little bit here, if N represents haploid, diploid is 2 times N, okay, because it's two sets of chromosomes instead of just the one. So if a diploid cell has 24 chromosomes, how many would a haploid in that same organism have? So in looking at this, you notice here's our diploid, right? We have 6 chromosomes, and here we only have 3. So meaning I have 2 sets of chromosomes and I just have one set of chromosomes here. And remember how you would find if N represents haploid and 2N represents diploid. You know here that you have 24 chromosomes for a diploid cell, but you're wanting to figure out how many haploid you have. So essentially, you would just divide this by 2, so N would equal 12. So when we go through meiosis, it goes through meiosis twice. And I'm going to talk to y'all more in detail about these um, phases when we're in person, but just to introduce them, there's something known as PMAT. And PMAT means prophase, metaphase, anaphase, telophase, and then cytokinesis. So the first thing that takes place is interphase. Okay, this is just kind of like a pre process. Once the cell goes through interphase, it then goes through prophase, goes through metaphase, anaphase, telophase, until finally it reaches cytokinesis, which is what splits the cell in half. So we're starting off with a mother cell, and then we're ending with 2 daughter cells. Well, that's meiosis 1. So once we go through meiosis 1, we go through meiosis 2, and we take those 2 daughter cells that were made in meiosis 1, and we go through the PMAT process again. Then we go through cytokinesis. So we started with 2, and now we have 4. So this breaks down what happens in each of the phases a little bit more. An interphase is where the chromosomes replicate and chromatin condenses. Prophase is where the pairing of homologous chromosomes occurs, and each chromosome consists of 2 chromatids. Crossing over produces exchange of genetic information. The nuclear envelope breaks down and spindles form. I'm not going to read each of these to you right now. Y'all can look at this, read it over, because I do think it'll be more helpful for me to show this to you in person in class. But familiarize yourself with this just a little bit so it doesn't seem like a total foreign language to you when we come together in person. But notice here, this is showing you meiosis 1. We get our 2 daughter cells. Now we're starting meiosis 2, and we're ending with the 4 cells. So I know that you heard me refer to crossing over, and this is something that happens during prophase 1. And what happens here is the arms of chromosomes exchange reciprocal segments of DNA, and this creates genetic recombination diversity, which is a good thing. This is what leads to the diversity that we all have genetically. So looking at this, here's our 2 chromosomes, okay? Now, parts of them cross over. We have this segment cross over with this segment, and when crossing over takes place, it literally trades that segment of DNA with the other. So you may ask, okay, yeah, I see that there's a genetic diversity here, but how does that change? How does that lead to more genetic diversity? Well, it changes because as this happens during crossing over and as meiosis continues, you are provided this variation because those pieces of DNA have traded with each other, causing diversity. But then as you move on through the process, it continues to share and to pass that on. So what started with one, we move on, and now we have all these different possibilities.